Let's see now the organic molecules. And organic molecules, it's all about the carbon. These organic molecules, usually they have a huge amount of carbons within their structure. And there are several characteristics of carbons. I will skip them. They are pretty straightforward. You can just read them. Carbons, as you can see, it can bind in single covalent bond, double covalent bond. It can bind to hydrogen, can bind to nitrogen, can bind to oxygen. It can form a long chain. It can form a linear chain. It can form circle, and so on. So all of these different forms of how carbon can interact with other atoms make it very fit to build life. Now, these organic molecules would come in simple forms as here, and these simple forms can be joined together to form longer forms, okay, or to form larger structures. Now, when two, like in this example, these are simple sugars. When we can join two simple sugars together by removing water from both of them. One of them will give the OH, the other one will give the hydrogen. We provide some energy, and we will get disaccharide. And we can add to the chain again by repeating the same thing. We will remove one hydrogen, add energy, or use energy to establish this connection, and eventually we will get long chain of carbohydrate. This might contain 1,000 or even 10,000 of these simple sugars. So this reaction, when we are removing the hydrogen from two starting molecules, is called dehydration synthesis. Dehydration because we are removing water, and synthesis because we are making something larger than the starting material. Here we started with 1 and 1, we ended up with larger one. Here, large one and small one, we got even a larger one, and so on. So this is a synthesis we are making, and it's dehydration because we are removing water. Now, if we want to break this, then simply we will reverse the process. We will add water and we will release energy. Adding water would break these structures into simple forms, just reversing the reaction. Adding water and energy would be released. Add water, and the energy would be released. Add water and the energy would be released. That is hydrolysis reaction. That is a hydrolysis reaction. Okay, let's talk now about, there is this video also, please watch it. Let's talk about some of these organic molecules. The first group is the carbohydrates, and the carbohydrates are used for energy and they are used for structural support. We have some monosaccharides. We have glucose, fructose, galactose, ribose, and deoxyribose. And then we have the disaccharides when two simple sugars came together. Like the sucrose is the glucose and fructose, maltose is glucose and glucose, lactose, glucose, and galactose. I'm not really interested in you knowing what forms what. Just be familiar with the names, okay? Just be familiar with these names. And also, I'm not really interested in you knowing the chemical structures, okay, or how the reaction is happening. Just understand that we can start with simple sugar through dehydration synthesis. We can get disaccharide reaction. Now, I want to show you something here in the ribose sugar. Now, the ribose is different from the glucose because the glucose contains 6 carbons. 1, 2, 3, 4, 5, 6. Now, only this C is shown because here the corners, this is the way the scientist approved on how to write, and instead of writing C, 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 so they will refer to it as corners. So the 6 carbons it means that the carbon is made out 6 carbon sugars. It means that the sugar is made out of 6 carbon atoms. Now, the other form of sugars that we are interested in knowing for this course is the 5 carbon monosaccharides. Here they are 5 because 1, 2, 3, 4, 5. And we have two forms, ribose and something else called deoxyribose. Both of them are 5 carbon sugars. Now, the difference is this oxygen. This oxygen. In the ribose, it has this oxygen. In the deoxyribose, it's missing. And that is the deoxy. Keep this in your mind. We will see it later when we talk about the DNA and RNA. Okay? So just understand that the ribose and the deoxyribose is missing an oxygen, whereas the ribose, it has it. I'm not really interested in you knowing exactly, the chemical structure of it. Just understand the difference between the two. Polysaccharides, when two disaccharides or more, they start to merge and they will give us the polysaccharides, which might be branched, and we are talking about sometimes 10,000, 100,000 of these monosaccharides. So these are going to store energy like starch or glycogen in our body. Both are stores energy, or they will give structural support like cellulose. This will give structural support in plants. And these are like some forms of them. See, these are polysaccharides, and see they can be joined together, branched, to give either structural support or to store energy. Lipids is another group. We have three classes. We have triglycerides, phospholipids, steroids. Triglycerides also stores energy. Phospholipids, they will form cell membranes, and steroids, these are let's say these are the hormones in our body. Triglycerides, they are made of fats and oils, and we have two different types of fats. We have saturated and unsaturated. This is a question for you. Try to know what is the difference between them. What does this all single bonds between carbons or include some double bonds between carbons? Try to find the difference and post it also in the forum. As we said, the triglycerides are to store energy. Phospholipids, their primary function is to make the cell membrane. And phospholipids, they are unique in their structure because they have a head, and that head is polar. Remember, polar, it means it has two poles like the battery, one positive and one negative. And they have non-polar tail. Okay, so the polar head would be hydrophilic. The polar tail will be hydrophobic. It will stay away from water, whereas this will meet water. This structure is extremely important in forming the cell membrane, and this is something we might see later when we talk about the cells in chapter 3. Again, you are not required to know the detailed chemical structure, just understand the polar head and the non-polar tail. The polar head is made out of phosphate group and the glycerol group, and the non-polar tail is made out of fatty acids, two fatty acids. Steroids, these are composed of 4 carbon rings. Example of them, cholesterol, as you can see, 4 carbon rings. This is cholesterol, and variations of it will give, for example, estrogen or testosterone. These are the hormones. Just know that these are, they perform specific functions, and their backbone is the cholesterol.
Triglyceride.
Proteins. Proteins are extremely important because they are the units that performing functions in our body. Think of gas exchange, okay, muscles work, white blood cells that defend us, and hormones, and many other functions within our body, all are performed by the proteins. So these are complex structures, and the basic unit of them is called amino acids. And we have 20 different types of amino acid. Each one of these amino acids, each one of these amino acids, it has an amino end that is contain nitrogen, carboxyl end that contains this group, and an R group. Now we're not going to go into the details. The video might show some details about them, but for me, just understand that the basic structure for the proteins is made out of amino acids, and we have 20 different types of amino acids. Now, when these amino acids join together, as we saw earlier, by dehydration synthesis reaction. So two amino acids will we will take H2O out of them, and we will get dipeptide. If there is a third amino acid, we will take H2O out, and the three together will be joined, and so on. And eventually we will get again 100k chain of these amino acids. A repetition of one of these, or different types, actually, not one, different types of these, amino acids. And these are the proteins. Now, the proteins, when they formed, they will have a 3D shape that makes them functional. This 3D shape depends on temperature and pH. So any changes in temperature or pH will make it unfunctional. And that's why it's extremely important to maintain the temperature and the pH. If the temperature or pH changed, that will change the 3D shape of the protein, and that is something called denaturation. Denaturation, and this will lead to loss of biological function. Think, for example, of when you are boiling an egg. If you break the egg, it's like it has the fluids, right? But when you boil it, they will become solid. These proteins, they were denaturalized, so these are not functional anymore. This is like a classical example of denaturing the proteins. Enzymes are subgroups of these proteins. So enzymes are proteins, and they play a critical role as a biological catalyst. What does that mean? It means that they speed up chemical reactions. Think of oil and water. These usually don't mix, right? But if you provide some catalysts, you might dissolve oil within water. Think, for example, soap detergent. Soap detergent is going to surround the oil droplets, and that's why we will be able to wash the oil or the fat with water. So the enzymes are going to speed up the chemical reaction. By the way, if you leave oil and water like 1 million years, they will react. They will say, okay, I think none of us is going away, and let's react. But that is extremely long time. So we will use the detergent to clean them up and to speed up that chemical reaction. Now, the beautiful thing about enzymes is that they are not altered or consumed by the reaction. It means that they can finish one reaction, like here, and go again and finish another reaction, and go again and finish a third reaction, and so on. Because enzymes, they don't get into the chemical reaction. They are just providing a space for the reaction to happen, to make it faster. But technically, they are not interacting with the reactants and no parts of them would get into the product. They are just providing the medium. I usually say think of, eBay or Craigslist. These are websites that someone wants to sell something, they will put it on eBay. You want to buy it, you will go to eBay. So eBay will provide this space that you can meet with the seller or with the buyer. But you are not taking any part, or eBay is not taking any part within this interaction, right? So this is the enzyme. This is the enzyme. This is the enzyme. The enzyme is not altered or nor it changed with this chemical reaction. And as we said, since these are proteins, so they are subject to the pH and the temperature. If the temperature is low or high, or the pH is low or high, these proteins, these enzymes will get denatured, and they are not going to be able to perform their functions, which might interfere with the life. The other group is the nucleic acids. And these nucleic acids, they are long chain of subunits known nucleotides. And we have two types of nucleic acids, DNA and RNA. DNA is made out of the deoxyribose sugar, whereas the RNA is made out of the ribose sugar. DNA is the that's deoxyribose nucleic acid, whereas the RNA is the ribonucleic acid. DNA contains information or instruction for producing RNA. Technically, it contains the genetic information that would determines who are we, like our eye color, our hair color, our body type, our our body mass, our muscles, how our skin color, and so on. Everything in us is determined by these instructions in the DNA. RNA would take a copies of these instructions and use them to produce proteins. Since the proteins are going to do the work, responsible for making the work, so we need like a guidelines or instructions how to make them. These guidelines or these instructions are coming through the RNA, which is technically a copy of the DNA. So proteins, as we said, direct most life processes, and the process would go like this, or the way of, or the relationship between them is like this. DNA will make a copy of it into the RNA. RNA then will be converted into proteins. So what are the nucleotides? Each nucleotide will contain 5 carbon sugar. It's either the deoxyribose or the ribose in DNA or on RNA, nitrogen base, and phosphate group. So this is the ribose sugar or the deoxyribose. This is the phosphate group, and this is the nitrogen base. Ribose, phosphate, nitrogen base, phosphor, deoxyribose, nitrogen base, and so on. So each one of these is a nucleotide. And for DNA, we have 4 types of nucleotides. We have adenine, guanine, cytosine, and thymine. Adenine, guanine, cytosine, and thymine. And these are they. Adenine, thymine, cytosine, guanine. Of course, I'm not requiring you to know these chemical structures, okay? But know the names. You need to know the names. You need to know the names and also know the pairing, that adenine always going to pair with thymine. Guanine is always to pair with cytosine. What does this mean? DNA is a double-stranded molecule. So we have 2 strands, and these strands are meeting each other. So on these strands, we have the phosphate group, the sugar, phosphate group, the sugar, they are lining on the floor, let's say, and the nitrogen bases, they are stretching upward or toward the middle. So they will pair together. And as we said, the pairing will be T to A and G to C. Or of course, AT CG. It doesn't matter. But it's going to always to be like this. Okay? So if you have a sequence, let's say, of A, T, C, C, G, C, I need you to think, to pause the video and think how it will pair, or what is the other chain would look like. So pause the video and try to figure it out. Okay, I guess you got it correct. So this will be T, A, G, G, C, G. This is the pairing. Okay? For RNA, it's different in two forms. It's single-stranded, and its nucleotide, it contains ribose. Okay? This actually, it's different in three ways. It's single-stranded. It contains ribose instead of deoxyribose, and it has uracil. It has uracil as one of its nitrogen bases. Adenine is the same as the DNA. Guanine is the same as cytosine, as DNA. Cytosine is the same as DNA, but uracil is different. In DNA, we had adenine, guanine, cytosine, and thymine. In RNA, we have adenine, guanine, cytosine, and uracil. So this is the difference. This is one of the differences between DNA and RNA. The pairing in this case would be A to U, or U to A, G to C, and C to G. Okay, but this pairing is not something we are going to see it usually. Okay, because most of the time, or at least in our course, what we are going to see, we are going just to consider that this is a single-stranded molecule as this. So technically, there is no need to know the pairing for the RNA, but it's again the same structure: phosphate, ribose, and the nitrogen space. Phosphate, ribose, and the nitrogen space, and so on, and it's a single strand. The last thing we need to understand is the ATP, or the energy molecules. These are nucleotide adenosine triphosphate, and this is a universal energy source. It can be broken down to give ADP plus phospho group and energy, or we can reverse the reaction and save the ATP. What are we talking about? Sorry. This is the adenosine. This is the this is the adenine. Okay, ribose with the phosphate group. Now, for this adenine, we will add 2 phosphate group, so we will have 3 phosphate group, and that's the triphosphate. Okay, so it is adenine triphosphate. This is the ATP. Now, the energy is stored here and here. We can break this phosphate group and release the energy out. How do we break it? By dehydration, by hydrolysis, by adding water. So we will move from ATP, an adenosine with 3 phosphate, into an adenosine with 2 phosphate, ADP. And we will release the energy. So the energy is out, water is in, we will release energy, we will release 1 phospho group, and we will end up with ADP. So when we are breaking ATP, we will get ADP plus phospho group plus energy. And that is how we moved from here to here. If we want to get it back, if we have extra energy, we have extra phospho group, we have ADP, we will reverse the reaction. And that is dehydration synthesis reaction. Dehydration synthesis reaction. We will put energy, we'll take out water, we will put the hydrogen, the phospho group, and we will end up with ATP. So ADP plus B plus energy will give us the ATP again. So whenever our body needs energy, it will use these ATPs. Whenever the body is making energy and wants to store it, it will use this reaction. Okay? I believe that's it for this chapter. Please post your questions and comments on the forum, and I will see you in the lab.